All right, thank you. Thank you so much. Yeah. All right, so thank you so much. My name is Humphrey. I'm a PhD student in the Biodata Analytics and Computational Genomics program. And then today I want to introduce us to Biomni for biomedical research. Myself, I've experienced this. I've used this for, I think, 4 months now. And then this session, actually on Agentic AI, has actually been running for about 4 months now. If you can see our GitHub repo, been having these sessions here, how to use Claude for agentic, for computational biology projects, how to use Codex, and then we did some other things with Phylo or Biomni. All right, so this is a repo that is freely available. You can check out some of the things that we've done in the past weeks and months. All right. So here we have a website here for Agentic GHX. You can visit the website, check out some of the problems we have, some of the things we are doing. If you want to learn some things, we have put together some concepts. We are still upgrading this so that people can have free access to AI and information about how to use AI,
Oh,
not only for.
sorry, Humphrey, we cannot see the slides. I think something, yeah, we cannot see the slides anymore.
How about now? All right. So,
Yeah,
so
now
I
we can
think
see.
I
Sorry.
think you can
Yeah,
see it now. Sorry.
thank you.
Yeah.
Thank you so
So
much. Yeah.
yeah, like I mentioned, we've been having a series of sessions on Fridays for the past 4 months on agentic AI for computational biology, and today is actually one of the sessions. So we actually advanced our tutorials, our knowledge sharing opportunities and exercises over the course of these 4 months. But then we thought it wise to revisit some of the concepts. So um, in the past 3 weeks, we had Samuel who took us through revisiting agents. All right, so this is also available for us to explore. And then I am going to build on some of the things that we explored um, just in the past month, July. All right, so I was mentioning that we have a website where you can find so much about us, our events, some of the things we are building, we are solving some problems, you can find them. We are actually making some opportunities available for fellowships, hackathons, and all that. So definitely, we're likely to have a hackathon that maybe people can decide to leverage Biomni, Cloud Science, or any other platform to perform some specific roles and all that. We also have some products and services that we are rendering or we are working with or ready to support with collaboratively or any other means can be discussed. All right, so back to my presentation. All right. So today, I want us to start by trying to understand what models are. We are trying to understand some fundamental principles. So we talk of a model as a system. All right, so a system that actually receives an input, processes that input, and then gives an output. That's a layman description of what a model is supposed to be. All right, so let's say, for instance, you have a data input. Let's say an image data. You have a model that is actually developed or built for maybe processing image data, or even if you take any model at all, like freely available model, you are likely to train this model to work on the data you are bringing in. And then based on the interactions or the different data points, the diverse and varied patterns in the data, this model is able to get trained to make subsequent predictions. All right. So that is what you should know about models for people with a biology background without so much computational biology or computing programming expertise. All right. So last 2 weeks, we explored foundational models. We actually went to see how we can also download some models and do some stuff we are still going to continue that exercise. All right. So we downloaded some of the models, and then we are looking forward to train some models, fine-tune some models, and see what we can be able to do by ourselves as individuals. All right. But in foundational models, we establish the fact that we think of foundational models just like any building structure that is put up for you to be able to actually guarantee how long the building is going to last or how firm is going to be kept. How safe the building is going to be dependent on the kind of foundation. And also, the foundation of every building actually determines the number of floors or the kind of weight you're going to be putting up, stacking up onto, yeah. Yeah, so foundational models or foundation models are models that actually are trained on different types of data sets. So here I was saying that if we have maybe an image data, you have a model that is supposed to help you do some prediction. So here for a computational biology example, let's say we have a protein model, okay, a model for maybe protein analysis, prediction, structural prediction and everything. You need maybe an input data, maybe you have a protein. You need a model to actually work on trying to predict the protein structure, and then it gives you the output, which is going to be the desired output you want, the protein structure or whatever it is. So a foundational model in this sense is trained on like different data sets or varied data sets. And then because of that, it has the ability to work on maybe image. It has the ability to work on maybe sound or voice. Data is having the ability to work on so many things. So we explored some examples. We listed Claude as a good example, ChatGPT, Gemini, and some of these others are foundational models because they can be leveraged to actually perform a specific assignment. All right. So they are not really trained for one specific task, but they can be leveraged. So you can leverage Claude or Claude models to be able to maybe do some voice analysis, maybe visual video generation. We can use it for maybe bioinformatics analysis. We can use it for maybe DNA mutation analysis and others. Okay, so um, this is what foundational models are. Now, when we are talking about agents, okay, so the agents are able to leverage models, and then they're able to break down tasks into smaller chunks, delegate them, and then they have the ability to leverage so many tools or platforms, and then to be able to give a very good output based on um, the user's desirable outcomes. All right, so let's take for instance, maybe I want an agent that is able to connect to maybe protein databases, and then is it going to download the protein for me, the protein structure from me, and then with that, is going to also help me profile the mutations, look at the structural changes. When it's done, I need the agent to be able to use the models available to be able to maybe give me a manuscript of the analysis that was done, put together the abstract, the introduction, the metas, everything, and then also give me a PowerPoint presentation if that is what I want. And then, yeah, so this is an agent that has so many things embedded in it. It has access to databases, it has access to softwares, it's able to perform so many calculations, computational tasks, and then gives you an output. This agent is also having the ability to try to take it, take any given task through several processes, putting them into steps, getting a plan such that it's going to follow suit as to what specific task has been executed before moving on to the next. So you can put in a loop such that it's going to confirm everything before giving you a final output. All right. So here we are taking Biome as an example. All right. So Biome, all right. Sam, please can you help me with that? So we are going to be using Biomni here as an example for our demonstration and yeah, our session today. So this is how the Biomni platform looks like. All right. So you can actually browse Biomni. When you browse Biomni, what you're supposed to see, let me open this in incognito and see how it shows. So when you browse Biomni, you're supposed to get to this landing page, the home page. All right, create an account. Once you have an account, you need to sign in. But before creating the account, it's going to ask some very good questions about who you are, the kind of research you do, the kind of analysis you do, and you're going to set some preferences as to how you want your outputs to be. Maybe if you want your code bases or the codes, the scripts that are going to be run, maybe for data visualization to be done with either R or Python, you are going to set such preferences. And then if you want the output to be maybe an SVG output or a PNG output, you can set that up and all that. So if you're able to log in successfully, what are you going to see as this interface? So this is how the Biomni platform looks like. All right, so here, where we are currently is our projects. So Biomni gives you the opportunity to be able to create projects. Now, just like how you use ChatGPT and other AI platforms, where you create projects and then you organize your chats in that specific project. That is the same thing that we are going to be exploring here with Biomni, and or that's the same concept it is going to be following. So here I have this project here that I have created that we are going to be using for illustrative purposes. Over here, Biomni has a hub that is containing several skills available to Biomni. All right, so here we are going to explain what skills are. Okay, so when we take, for instance, this skill, for instance, a skill is actually a set of instruction, right, that you're going to hand over to an agent, such that the agent is going to specifically perform tasks based on a structured outline that you're going to present to it, giving it instructions, the do's and the don'ts and everything, what the agent is supposed to understand about this skill, the limitations, the output you're expecting. If you have maybe some installations to do, the skill is going to do that for you. So here, this skill is specifying that for you to be able to use it, or if you want to perform a specific analysis that is related to ancestry-aware GWAS fine mapping and annotation, you should first of all be expecting some inputs. This should be maybe some GWAS data sets or summary statistics. And then you need information on the ancestry. All right. And I think the 1,000 genome here is an example. Then you need to also specify some specific traits in the data set that you're looking at. And then maybe annotations, whether you want to annotate cells or and some tissues and all those things. I think these are some things that you would be needing for you to be able to do this ancestry-aware GWAS fine mapping. And then it's going to give you the desired outputs at the end of the whole analysis using this particular skill. So yes, you have the opportunity to be able to create your own skills for different scientific work. So here, let's say Biomni has an inbuilt tool for literature review. All right, so we can look at this particular skill. Yeah, so this is a skill for literature review, and then this skill is supposed to be used for audit-ready biomedical reviews that require exact quotations, table sentence or figure locators, claim evidence tables, contradiction checks, and all this. So this is quite very important for we computational biologists if you want to do a literature review. So this skill, it is presenting to the agent or to the AI platform that when you want to use this skill, use it only when the user explicitly needs one or more of these specific requirements. So maybe the person wants exact sources to sentences or a page, which is very, very common with literature review. Or you want to audit already claimed, or you want to audit something that you've already established, maybe you've already written. So you want to verify a claim to an evidence. All right. So you can do that. Maybe you want to verify citations and all that. If you want to also do a comprehensive report of all those claims and so many other things. So these are like sets of instructions that the AI is going to be working with. So you have inputs. So your inputs can come in a way of maybe a review question. You have Please write a review on how agentic AI can be used for computational biology in research institutions in the NIH or research institutions in Ghana or research institutions in KNUST. And then this skill is going to be provoked to perform that specific task. All right. So it's also going to help you write, like do a full text, something on your write-ups or yeah. So you are going to have an output here. And all these things are actually like output from sub agents that perform specific tasks, hand it over to maybe a major or a main agent to perform downstream synthesis. All right. So there are so many things that the AI is going to require from you based on this skill, just to get in-depth understanding of what you want to do. So it interviews you, it brings up some questions to find out, like, do you have specific PDF citation lists or prior claim table to use? If you have them, you upload them. Then maybe you want to say the review you want to do should be a quick review. It should be a deep review, or it should be a broad review. That can be done for us. All right. So that is just one of the things I need to talk to you about what skills are. So talking about skills, you can also maybe come up with your own skills. So let me close this incognito tab and then let's see. So you can create your own skills. This is an account I'm using, all right? So here I have my own skills. I've been able to come up with about 22 skills over here. So here, to be able to work with skills, you can find some skills online. Okay, so we can go online and then find some skills. These are some skills I'm going to recommend to people who are interested in scientific writing. There is this GitHub repo called Cloud Scholar. All right, so if you want to search for skills yourself, you can actually maybe come. I need a skill for bioinformatics. Skills for Claude. So definitely you're going to find some skills. I think there are some skills we can maybe explore over here on GitHub. Some people, okay, so Claude has this plugin for life sciences. It's something that can help you actually. So there are some couple of skills that we can explore, but let me take these two. So we have bio skills. That is someone who is generous to share this for us. All right, so this skill can help you with alignment. It can help you with ATAC-seq, can help you with causal genomics, chemoinformatics, ChIP-seq, and so many other things. All right, so this is going to be based on what you need. Maybe it contains skills that have maybe some specific things on genome assembly. So genome assembly here, let's see, we have maybe a skill for genome assembly polishing. So here, if you have this skill, what it means is that whatever AI agent you're using or whatever AI platform you are using can be able to perform genome assembly for you, especially genome. Here we have genome assembly polishing. So here it contains the set of instructions that are relevant. All right. So these are so many tools that we use for either long read or short reads. All right. And so many other things. All right. And then we have, yeah, you have scripts over here, codes that are going to be running provided you provide, you are going to bring in an input. All right, so we are going to briefly demonstrate that. So here, I actually went to Pathogen Watch. Pathogen Watch is a platform where you have bacteria genomes being collected and analyzed. All right, so over the years, they've expanded beyond Klebsiella pneumoniae to other species. Now you can check it out if you're interested in bacteria genomics, sorry. Yeah, so you can study that. But then I had to download a genome. I came here and then I had to randomly download one genome. So here we all know that what the genome I'm downloading is Neisseria gonorrhoeae. All right, so I have it on my computer. I've already downloaded that and I have named it unknown. So unknown.fasta is an assembled genome. All right, so here I want us to actually demonstrate something. Please kindly let me know if you can hear me. Um, Probably. So I get to know we are following, please. All right. So here we all know that we are working with this particular organism, but then I named the organism unknown. So let's see if we upload this to Biomni. The Biomni is going to be able to tell us what organism that is, and then we take it out from there. All right. But please, do we have any question up to this point before I continue? Do we have any question? All right. So if we don't have any question, first of all, when you open Biome and you want to start maybe a project, you can maybe create a project first of all to say, okay, this project is about maybe I'm coming to do bacteria identification, but then I have created this project. So once you hit create, it creates for you. So I created Agentic GHX demo. So here, what we are going to do is identify this organism. So here we just have to type in plain English, just like what you do in ChatGPT in your chat box, or Claude, or Grok, Gemini, and any other AI platform that you're used to. You just have to type in English, and then because of the agents, the models that we have, they're able to pick up this, and then the skills we have. Yeah, since uh, since we have several agents that are going to execute this, they're able to just translate all these things. If we need to do some computation, some calculations and all that, the specific agent, the specific skills are going to be employed. All right, so identify these organisms here and um, and suggest downstream analysis I can do with this, yeah, with this organism. So by explaining this, what I am trying to say is that people can actually upload files or even a whole folder of any data they have, and then it's going to help us, uh, sorry, come to upload our genome or any file that you have, any data sets that you have, you can just upload it here, and then Biomni is going to continue the analysis. So as we can see, this has been uploaded, and over here, we see the upload showing. So we can just go ahead. I want to use the standard model because I want to conserve some usage credits. All right, so this is going to run for some time, and then we see how it goes about the work. So, in using our AI agents or our AI platforms, our AI tools, one of the things that we need to do, especially if you want to brainstorm ideas, if you want to perform some analysis using AI, you need to first of all plan. So you have to allow the agents, okay, plan how they understand the question you are posing or you are presenting, and then they are going to give you a detailed outline of how they are going to go about executing the task. So here we see the agents trying to read the file that we've uploaded here, and then it's going to analyze, perform several processes. So here it is going to use all the available agents that are necessary for this. And then we see here, organism identification. Oh, sorry. So what I downloaded was SARS-CoV-2. I'm sorry for that. All right. So what I used for, all right. So what I used for this particular one um, was the Neisseria gonorrhea. All right. So what I used for that one was Neisseria gonorrhea. I just wanted us to use, uh, sorry, this is it. All right. So we can see what I used previously, or I need to see this genome analyze gene expression. Okay, first file sequence. Yeah. So what I did previously using the Neisseria gonorrhea, I named that FASTA file Biomni and uploaded it. So I said, look at this file, analyze this data for me, identify the organism, and profile the genes, and visualize the results for me in a PPT slide or a PowerPoint slide. So you see, it went about giving me a plan. So we'll come back to that in a bit. Let me just get to what we have. So here, it means the unknown FASTA that we have here was a SARS-CoV-2 genome, which was another genome I downloaded from Pathogen Watch, like I showed you. So you can go to Pathogen Watch, find the organisms, and then, yeah, you have SARS-CoV-2 here. That was what I downloaded. All right, so it's telling us that this is a SARS-CoV-2 genome. And then this, the genome length. Okay, this is the GC content. It has calculated all these things for us. Now, what I want to mention clearly is that this AI is actually linked to an HPC. So people who are not having computational resources, but then want to do some of these analysis, you should know that this is going to be done on an HPC. So there are several plans to this particular there are several plans to this particular account. If you create an account for the first time, you have 3 days pro trial, but then if you exhaust that, you would have to actually start paying for the pro plan, which is $100 per month. All right, so that is another thing I need to mention. All right, so back to this. The organism had been identified. Biomni is telling us the organization of this genome, is giving us some specific traits, some specific gene contents or specific regions of these particular viral genome. And then, from the question, I said suggest downstream, subtypo, downstream analysis I can do with this organism. So even with that typo, it gets what I want to do. All right. So here, it is going to suggest to me specific analysis I can do. Maybe I can do a variant analysis, or a lineage assignment. I can do maybe protein annotation, or if prediction, I can do structural biology and all that. So depending on whatever analysis I want to do, I can so we have a lot. We really have a lot we can do with a single genome. So this is something you might want to explore. But then I want us to also try out something. Let's say I want to explore a research gap. Sorry, a research gap in antimicrobial resistance using the um, um, okay, using the recent outbreak of um, cholera in my area. Okay. So I want to do this for maybe a scientific conference or something. Let's see what Biomni is going to do. So first of all, this is like an ideation process. This is not like a final input and instruction to do something. So Biomni will have to, or the AI would have to think about this and then ask me further questions, all right, to find out exactly to a very great percent or a large extent to what I want to do with this particular idea. So here is saying before I proceed, I'd like to clarify a few things. One, so that's under outbreak and then the data. What is the cholera outbreak you are referring to? And do you have any local data, whether clinical isolate sequences, susceptibility results, epidemiological records from it? So I don't have any of them. All right. So no local data yet. Um, so here I want to say let's pilot with public data. So what is the angle of my research? Which AMR research angle interests you most? So either I'm interested in genomic surveillance, resistance mechanisms, or treatment. I think let's go for transmission and One Health. So what are the outputs I'm expecting? What are the deliverables? So maybe I need a literature gap analysis, or I need a study protocol design, or maybe I need data analysis, I need a grant, maybe. So I have to choose one of these things. So here, let's explore literature gap. And then what is your, okay, it says my role and then the resources I have. What is your role and resource level for this work? So here I'm a researcher, but maybe I think computational focus. Yeah, yeah. I don't want us to do a wet lab aspect. So I have to submit this. Now, when you submit this, automatically, um, Biomni plans everything. So I told you initially that if you want to use the AI for a specific task, it's good for you to give a prompt and ask the AI to plan how it's going to execute the task. By doing so, the AI is going to actually go through a very rigorous step end to end to see the possibilities of the things you're asking for, the specific steps, the whole workflow is going to be outlined, the inputs you need, and then the desired outputs you are going to be expecting at the end of the day. So it puts this together for us. And then once you are okay with it, you approve of it. The reason is that you're going to be using so many tokens, using these AI platforms, and then you might not just want to start running something to maybe to a point where you are exhausting your tokens or your usage limits. Then you realize that no, I wish I had done this in a different way. Okay, that would have been a waste of time, waste of tokens or resources. So here, Biomni by default is going to plan everything. Okay, so that is what I think we should take from here. If you are prompting, especially for computational biology or anything, try to get the AI to understand what you really want. It gives you an outline. So here it's still asking for some information because it needs clarity. That was a very broad and a vague request from the AI. All right. So here it needs to narrow down all the things I need. So here I want it to be focused on Africa, and then the gap we want to explore should be maybe okay, all One Health gaps. Then yeah, how deep should the evidence retrieval go? So should it search online for abstracts only, or should it search for full text papers? And then, yeah, what do we want? So you can actually ask the AI to decide if you want. Then the deliverables, is always important to tell the AI the deliverables you're expecting, the outputs you're expecting. You give the AI an input and you're like, for this, I am expecting maybe a Word document, I am expecting an HTML, I am expecting maybe a PDF and all that. So you have to specify that. So here, maybe I need maybe a report. Uh, Mind you, here we are choosing a literature review report, a literature review gap. So a literature gap. So I think I'll need a report, if possible, a table for that, and then a PDF. But then let's do something that is not going to take so much time. So let's do reports only. So this AI takes it again and process it, and then it's going to find out if there are some additional information you would need. If you need some extra information, it brings the prompt back to you to select yeah, those instructions, and then it goes back again. If everything is fine, the AI is satisfied, it goes ahead to give you a plan. So now we would explore some of the things I have done previously so that we can forge ahead. Please, do you have any question? I think someone was unmuted. Do you have any question? Let me check. I think we have some questions in the chat. All right, some comments. All right. Thank you so much, son. All right. Please, do we have any question? If you have any question, kindly feel free to unmute and then talk. I'll be happy to do that along the way. Let's not wait till the end. All right. So here, Biomni is done. It has given me a plan. All right. So this is actually the plan Biomni is giving me. I have to read through this plan and see if I am satisfied with everything that is in it. Once I'm okay with it, I'm going to say yes, proceed with the plan. All right. So here, let's go ahead and read the plan. So it's saying that cholera, AMR for health literature gap analysis. This is focused on Africa. So the summary, so this is going to be a narrative literature gap analysis. Okay, so it's going to identify underexplored areas in cholera, antimicrobial resistance transmission, and One Health dynamics. This is focused on Africa, and yeah, blah, blah, blah. So this is going to give us the scope. So it's going to give us what the topic is going to be about, the geographical region we are focusing on. And then here we are interested in the gap. So the emphasis is like all One Health gaps equally based on surveillance, environmental reservoirs, animal reservoirs, transmission chains, mobile elements, wastewater surveillance, policy integration, and then, yeah, this not restricted to computational gaps. Okay, fine. All right, so we have to read through this carefully, understand everything that is going to go into this. So here, the approach that Biomni is going to use, first of all, it is invoking this literature review skill. And this literature review skill is supposed to put together all we need in so many steps. So here is saying that it's going to use the literature review skill. There's a specific agent for searching for literature review. So here is going to decompose what we are working on into focus subtopic queries. And this is going to be up to 20 papers each. Then after that, it's going to merge and then deduplicate. All right. So it's going to remove duplications in the papers we have, the hits we have from the DOIs that are, yes, that are going to pop up. And then it's going to normalize all these things. Then these are the plan, like under the step one, that is going to be followed carefully. So it's going to do all these things. For the sake of time, we have to go a little bit fast through this. So step two, it goes through that. Step three tries to synthesize the gap analysis and all that. Then the step four is when we are going to get our outputs. Mind you, in our outputs, we said we needed a report. So in this report, we are going to have an executive summary, we're going to have a scope, and then the research methods, evidence map, and all that. So here, the caveats here, I would want us to treat this as like a platform for learning, a platform for brainstorming, try to fine tune ideas and everything. You can be able to get maybe very good concept, very rich and yeah, top-notch ideas from this, and then you can do it manually. Yeah, if you want to see a draft from the AI to see how it's going about it as a template for what you're doing, you can also leverage this. All right, so we see the deliverables over here. They are going to be in-text citations throughout. All right, so here for the citation, it's going to still employ this agent to do some integrity checks. So here it's saying that it's going to cite only records that are returned by the citation, sorry, by the literature search agent. And then this citation, we can guarantee that to a very greater extent that is going to be from the articles that are going to be listed based on the agent responsible for pulling this literature from online databases or open access databases. All right, so once that is done, it's also going to mark which claims rest on the full text and then which ones are just from the abstract only. So imagine we had said that the review should be based on only the full text. I think that is also going to be streamlined. So I think this is quite good for us. And then, so let me just accept the plan. So this is running on the background while we attend to other things. So like I mentioned, there is an HPC on which Biomni is actually performing this task for us. So if you accept the plan, Biomni now puts everything here into a to-do list. And if you check from the slides, we mentioned that the agents actually are supposed to reason by breaking these bigger tasks into smaller questions or smaller tasks or jobs or steps, phases. And then there are going to be some tools that are going to be delegated to perform some of these tasks. And then we have some databases that are going to be consulted or made reference to. We have some softwares, so those who are used to bioinformatics, if you want to perform some of these analysis. So sometimes you can have Biomni saying that it's going to pull maybe a GitHub pipeline, it's going to pull a tool, install a tool, and then perform the activity. All right. So this is a robust platform for computational biology, any kind of computational biology. Whilst this is running, okay, through these steps, I would want us to explore some of the skills that are available. And then what I have on mine as well. Okay, so we have some skills here from Biomni. So currently, at the time we are having this session, we have 80 different skills that Biomni employs. All right, so we have single cell transcriptomics. This is a skill for data analysis. This is a skill for epigenomics, genomics and genetics for data analysis. This is for multiomics. We have microbiome analysis, very good, like end-to-end analysis. For those who are interested in drug discovery, we have skills for that. Molecular design, bulk omics, still multi-omics, bulk RNA-seq transcriptomics, yeah, drug discovery again. So many skills, so many skills. In fact, those who do wet lab can do this. I think I found a skill some time ago here on primer design, PCR primer design. So if you have primer sequences and you want to do some things, I think you can explore Biomni Lab for that. So not for those who do bioinformatics only, but then for others who are much more wet lab inclined, you can do primer design over here by just maybe saying, I have this paper, let's design this primer or do this. And then you have a whole set of instructions as well as the codes that are going to be used for the blasting and the primer design. It's going to plan everything and then do all the analysis you need to have for you to have some good primers for your work. So it's going to accept these inputs. If you have maybe a target DNA sequence in one of these formats, let's say like the FASTA file we uploaded, you can upload a file and then it's going to help you design a primer for that. If you have maybe some accessions from GenBank that you want to work with, if you have a sequence, maybe a sequence for a specific gene or a specific target that you want to design a primer for, you can do that. If you have a name of a gene and the organism. So I think this is quite interesting. Let's explore this for people. So let's say I want to design a primer for something. So I want to create a new task, design a primer to target things that can be used to identify this organism. So here we are talking about molecular characterization of maybe the organism. So we need to target, sorry. So I don't know any organism in mind. Let's come to genomes and explore over here. Organisms. Let me take an organism I don't know. Okay, I don't know this organism. So I think I'm going to copy the name and then let's come to Biomni. So this organism, uh, and then let's see if Biomni is going to do this. So just as simple as maybe bringing a question, a biological question, which you are typing in English. You have so many skills, you have so many agents that are going to be employed to perform some of these tasks. So briefly, let's check on our run and see how it's doing. Okay, so I think it's still, okay. So here you can see what the agent is doing, and it has to go through all these steps. And then if it is done, it organizes the results over here nicely for you. If you also want to see the computer resources that are being employed for some of these things, you are going to see. So largely see some Python, yeah, some Python functions are being employed for these analysis and all that. So I think time is really catching up with us. So here, Biomni is telling us that, so we are back to the primer design experiment. Here, Biomni is telling us that our design PCR primers for molecular identification of this organism. All right. Okay, so I didn't know that this organism causes kidney disease. Okay, so before it starts, it's saying that it wants to plan the approach, and this is very good. So I think if not for anything, um, some of us should leave this particular session with a mindset of that we are going to plan everything before executing maybe an analysis with our AI agent or maybe any query you want to do. Tell the AI, I want to do this. Now I need you to plan this particular thing, put it into steps on how you're going to go about this whole thing from end to end. And then it's going to do some of these things for you. Maybe if you're using Claude or some of the things, you can actually, I think Claude Science will be handled by someone else. Yeah, look forward. We are expecting a, a special guest very soon by faith. Very soon by faith. All right. So we are waiting for this to design the primer for us. And then you see that um no. So here I had a previous file that I've uploaded here. The AI is trying to infer from this particular project, thinking that maybe the file I uploaded could be that organism. So here you see there's a user uploaded FASTA file. Let me inspect it to understand the sequences and to understand what sequences are available. This could be the target organism's genome or gene sequences, you see. But unfortunately, that is not it. So he's saying that this is a draft genome assembly. It contains 95 contigs, space, assembly, whatsoever. Um, so, so the assembly, which is about 2 million bases, consistent with whatever strain. I think this is quite surprising. Let me quickly check this and see. So it's asking me for some questions. Which target genes should the primer be designed against? Yeah, Jane. You are coming here. All right, sure, join. Yeah, so maybe I wanted to target the system sRNA, or I wanted to target what kind of PCR am I going to be doing? Yes, I didn't specify this. So I'm going to do a conventional PCR. What specificity context matters most? Which organisms must the primers not cross-react with? I don't really know, but then I think both and also environmental bacteria. All right, so what are the deliverables I am actually expecting? So yeah, maybe you can have a primer table and then a validation of it too. And then maybe if you want a full report, it's going to give you that. If you want maybe primer table only, that is going to be done for you. So let's say I want to do the simplest tax here so we can be able to appreciate the platform. So while we wait for that to get executed, I'm showing you some of the things that were previously done. So here, help me. So this is something on accessing GUO data sets. So I had to type in plain English for the agent to actually interpret, understand what I need, deploy sub-agents, or make use of skills, make use of other connectors and all that to be able to work with this thing that I'm trying to do. All right. Now mentioning connectors and all that, I want to also mention that we have what we refer to as connectors. So connectors are like programs that help you interact with some other platforms or softwares or yeah, tools if possible. All right. So you can connect Biomni to your Google Drive such that when it's done with the analysis, you need to push it to your Google Drive, your Dropbox, or your box, whatever you have that you've connected. This is not connected. If you have maybe these are MCP connectors too that you can connect to GitHub. I can push everything, the repo that is going to be created, the results and everything into, I'm going to push it um, onto GitHub. And then if you have some custom MCP servers, you can actually connect them. I think Phylo doesn't really have so many connectors here, like maybe we'll have on Cloud and other platforms, but it's fair. So here, sometimes you can have APIs. So APIs are also a leverage to some platforms or some tools or workflows that you are going to use for your work. Okay, so by getting an API key, so thanks to Phylo, I got API credits for both. I think it was about $800 worth. Yeah, API credits from both to do a particular project. All right, so it's quite sensitive, the project, because it's still being done, and then I look forward to putting it out there in the earliest possible time. So you can get API keys that you can connect to different platforms. So both is also for protein structure predictions and some modeling. You can read about that, or those who are used to protein research are familiar with some of these things, or structural biology people are used to some of these things. If you have to make use of AlphaFold, Alpha Genome, and some other things, you need to get the API key and you can connect them easily. So yeah, unfortunately, exhausted my Bolt's API credits. So you can actually connect Biomni to other platforms to be able to do some of these things. All right. So, yeah, initially I talked about the personalization. You can choose the preferred language you want some of these things to be done, the analysis, the data visualizations, and all that. And then you're going to also specify the output format. Maybe you want either SVG, PNG, or any of them. All right. And then you can choose different teams to use and all that. I think that is all from here. All right. So let's check on this to see if our primer. Okay, so here it hasn't started designing the primer yet. It has given us a plan. So we have to read through the plan. Time is really, really against us. So we are going to read through this plan. If we are satisfied with everything, we just have to approve it. And then it's going to do everything for us. All right. So I was exploring something. Let's see. Okay. So um, I needed to do a project and so I was taking a neurogenomics, pharmacogenomics course this semester, but I need to do a mini project for both. I want you to suggest projects I can do with publicly available data. I have an HPC and I can possibly use Biomni for that. Help me brainstorm good ideas. They should be publishable eventually. Yeah. So I did this. It gave me a whole plan on what it was going to do. So first, maybe drug repurposing, TWAS, pharmacogene expression. Then for the neurogenomics course, Mendelian randomization, cell type specific heritability enrichment for a specific neuro trait, and then fine mapping, and then functional annotation using GWAS data. Yeah, so the AI helped me. So for two courses, I needed two separate projects because I wanted them to be publishable and all that. So here there is an interview or yeah, an interviewing here or further questioning here to understand to get the context right for what exactly I want to do. So this had been done. I had to choose some of the things. And then how about something variant calling and copy number variation works? So yeah, you had to. So you have to actually use platforms like this only if you have maybe a very good understanding of the research domain, such that you can be able to help guide the AI to do what you want to. But then, yeah, it's going to be risky if you are trying to do something and you barely have an idea or in-depth analysis on that specific thing. Oh, sad. So I unfortunately I ran this, but I can't find the results. I could have showed you some things. Uh, all right, so this actually had to be a detailed plan. Please, is there is there is there any question from someone? I think someone is unmuted. We can take.
Hello,
Yeah.
good evening.
Hi, good evening.
My name is Nicholas Nobo.
Okay.
Yeah, I'm from Uganda. I'm a third year biotechnology student.
Okay. Yeah, please.
Yeah, yeah. The technology is really good that you have come up with. Yeah,
Thank you.
but me, I have one question. Have you like carried out tests like like many people to log in like when you have very many users like totaling to 100 like very many people using the same technology at the same time, have you tried it out?
Um, Nicholas, kindly take that question again. I think I lost you at a point. So you are trying to say if so many people are using this technology at the same time, are you going to maybe have issues using it or not? Is that what the question is about?
Yeah, that's my question.
All right, yeah. So I think some of these AI platforms are really doing a very great job to have us use the platforms because so much money goes into maintaining the systems, the clusters on which they are being run, and so much money goes into even the cooling these machines is a lot of money, electricity, and all that. And then sometimes we need to compete for some of these resources. So mostly priority is given to people who are on the pro pro subscription. But then for the past 4 months that I have been doing um stuff with Biomni, I have never had any issue with trying to ask a question and then they'll say maybe wait, your work will have to be running later or something. All the time when when I come around to write something, I just have it running. The only challenge is on the free account, you just have a very short usage limit, but then it's very good enough for you to be able to run some of the things. And one thing I think Biomni has been so kind to do is that I realize when I'm running something and then my session ends, on the background, this keeps running. I hope they don't stop it. And yes, we are just pleading with them that they don't stop it so it can continue on the background until your usage limit resets. All right. So I observed in Ghana here, maybe every midnight, you have a reset for every day. But then if you subscribe, like I mentioned, today you are likely to have a pro plan that is going to be on trial for 3 days. Yeah. So I don't know if you are answered.
All right, thanks very much for the
Yeah.
reply.
All right. Thank you so much. So those interested in molecular docking, drug discovery, molecular dynamics simulation and everything, I think, yeah, Biomni has been able to do some of these things. You can check out, can you see my screen, please? Let me see. No, I'm not sharing. Yeah, so I think let me read some questions in the chat and let's see if we have everybody here. All right, so please what makes Biomni different from ChatGPT or other general AI agents for biomedical work? All right, great. So Jefferson, I think ChatGPT, Claude, and all these AI platforms, some of them are foundational models. ChatGPT is a foundational model. ChatGPT, sorry, has foundational models. Okay, let's put it that way. ChatGPT has foundational models. Claude has foundational models. All right, so when we're talking about these foundational models, these are models that actually give you, like, they are trained on different types of data sets. So you see what we are exploring. Somebody can do drug discovery, somebody can do single cell, somebody can do primer design, somebody can do bacterial genomics, somebody can do GWAS, TWAS, so many different angles, okay? I've been trained, okay, on, like, this model has been trained on these diverse data sets. And because of that, we can leverage it to perform a specific analysis. So today, if we don't have maybe a foundational model for something, maybe like, definitely we have so many foundational models. We have so many models for so many things currently in biology, tech, finances, and all that. But then I'm trying to say that if we take, for instance, a model that is supposed to just predict, okay, um, whose image this is from maybe thousands of images. That model is specific. So that's a specific model for maybe image prediction. But then you need a foundational model that can perform several tasks. When it performs some of these several tasks, you can narrow it down that, okay, if you are able to predict images, I want you to tell me what these images in cancer. Okay, so it's like you are trying to leverage the capacity of the foundational model to do some image prediction in maybe cancer cells imaging or brain tissue imaging and all that. All right. So ChatGPT and all these things can do this, but you can't leverage Biomni to say that, okay, Biomni, you have this. I want to maybe also train a model, or I want to come up with a model that is going to do brain imaging analysis if Biomni doesn't have that already established. All right, so Biomni now is like an agent that is performing biomedical research assistance. Okay, so that's the difference. Biomni is specific for biomedical research assistance, but then it leverages so many models. Like we have the max, they have their models that have been classified as the mass, the fast, and then the standard, but then you can use custom models. I don't know if you're seeing my screen. No, let me share my screen. All right. Yeah. So over here, on Biomni again. So we have some models here that can be used for any task you are working with. But then you can use some of these custom models if you want to use Kimi 3, if you want to use Claude Opus model, or the GPT 5.6 model. You can decide to use this model to do the task that Biomni has described. So the agent is performing only the instructions, only the things that have been built on Biomni to perform. All right. So that is the difference. I don't know if we have another question in the chat. Unfortunately, I didn't see them come up. Oh, I think someone actually answered. Okay, so can one use this AI agent for systematic reviews? Yeah. I think you can use it, but you need to be guided as to how to go about it. It's not going to be a very good one for you, I think, because it will have to come with some level of prompting, or you have to maybe come with some skills if systematic review skills are not developed here. All right, or they are not available on are not available on Biomni. Yeah. So do you have any questions, or we can just go do something? So yeah, I think I wanted to show something, but unfortunately I'm not finding it. I think one of the things we are doing, the cholera work is done. You know, we were writing, we are trying to explore some research gaps in AMR using recent outbreak of cholera in my area. All right. So we said here we were going to do a narrative literature review to explore the gaps. So this is the output from here. So we have an MD file here, okay, on the gap. So here, antimicrobial resistance in cholera and One Health gap analysis would focus on transmission dynamics in Africa. All right. So it gives us an executive summary, the key findings and all that. So here you can go ahead to tell it to maybe put it together into maybe a proposal or a review or something. So this is actually going to be so helpful if you need to brainstorm some ideas around something you are thinking. You can actually find like very good stuff, and then there are in-text citations. Yeah. So if you want to look up for the references, the citations yourself, you can tell Biomni, and it's going to pull up the references for you into maybe an Excel file for you, so that you're going to manually do all these things. Yeah, so that is one of the things this can do. What other thing were we doing? The organism identification. All right, so identifying the organism, we said here it was going to give us a plan. I think we didn't do any other thing after it gave us the plan. So yeah, so I think, yeah, today's session will be brought to an end if we don't have any other questions from everyone here. All right. If not, then we have to probably please. All right. So here, there was something I was doing with GWAS. I might briefly show you this. So okay, so if you see the to-dos, here it was going to set up some compute environments, and then here it was also going to help do a PhD proposal document and also build a 9-slide PowerPoint from these GWAS. I just want to show you how simple the interaction has been. So review this research that was published here and here. I need to suggest potential follow-up analysis I could perform using GWAS summary statistics, variant, and or variant files, particularly in the context of a PhD level bioinformatics research, include ideas for downstream functional interpretation, integrative genomics, and novel hypothesis generation. So this is what I came to ask, and then it gave me an overview of this particular work or research, a very robust GWAS study with over 800,000 participants. And then he put all the things into a very good plan for me. He told me all the things that he's going to need to perform these tasks, the RAM usage, the number of cores I'm going to use, and then the runtime for each of the steps. So you see, Biomni is even able to tell me that based on these kind of things, these are the resource estimates I'm going to be using. So you can run this analysis and think that, okay, I want to do this in maybe one minute, 1 hour or something. So when you accept the plan, it gives you like the interrogative questions or that kind of interviewing it for the best instructions you would want to work with. Once that is done, you approve that plan, and then it starts executing everything. So he started executing everything, and he told me that drug target has been actually delivered to me. So here under the results, I told you that all the results will be put together here. So for this particular study, I worked with the both credits I got from Phylo. So if Phylo is hearing, Phylo should give more credits. All right, so here are some drug targets that have been actually identified and everything. If you want some of these things to be done with maybe a fewer summary, phenotype stuff, gene prioritization, ancestry analysis, like from the European data, the few African represented data, and all that, all these analyses were performed for me. And then I have uh, my genes related to this project was related, it's actually a dementia project, um, all cause dementia, vascular dementia, sorry. So these are the genes. Please, do you, Stephen, are you saying something? Okay. So after this, I had a report, okay, of the follow-up analysis report I can do about this. So you see, the interesting thing was I just had to pull a DOI and say, what are the further analysis I can do with this? And then it's done it for me. And we even had to do the project. So just like an end to end, all right? So you need expertise to be able to validate some of these things that you are doing. Unfortunately, we don't have access to computing resources or computer resources. So yeah, Phylo gives us the opportunity to work on their clusters, their HPC, and I think that's so kind of them. All that. But then the caveat here is that you would have to upload your data to Phylo. So I'd encourage you to read on Phylo's frequently asked questions, and then find out all the things about data protection and yeah, the data safety on their platform. But then I read it, and then to me, I was comfortable to bring in data to that platform. All right, so I asked for a presentation slide as well. So here I put together a PPT slide for me if you're not comfortable with it, you can modify it. So here I have beyond this, these are some analysis that I can do. I have several pillars, the functional characterization. And you see, the figures that were generated from this particular analysis were embedded in this presentation just to talk about some of these things. So if you're not comfortable with how the PPT looks like, you can just interact with the agent and say that, please change this PPT. I don't like the dark background. This is an academic presentation, and maybe I want this, this, and this. You have to prompt it. So please, if we have no other questions, I think we will draw the curtains here, and then we would, we'll have a wrap for this week, and then we come back here next week for another session. All right. Please, do you have any question? Any question? All right. So if we'll,
I guess a big, question someone will ask is that, um, like how do you, if you are maybe like a student, maybe in undergrad or maybe PhD or master's, starting out
yeah,
afresh with no data, how do you get started? And I think it's a very genuine question for
yeah,
most people, especially starting research like afresh,
yeah,
with very little support. So and I think this is a general question probably to everyone, but and I'm sure maybe some people are facing. But how do you get started now with these tools that can do so many things for us, and you are just really starting. How do you do it?
All right. So what I can say, first of all, giving this a shot, is that Phylo or Biomni is actually connected to so many databases. All right. So they are publicly available data. Phylo can have access to GenBank. Phylo can have access to PubMed. It can have access to NCBI. Um, I'm not sure about NCBI, but then I mean maybe CCGA, the Genome Atlas for cancer. It can have access to that. So many databases. Just trying to see where those things are located, and then I can share. But then if Phylo is able to have access to some of these databases or a lot of these databases, then what it means is that if you don't have data, you have a concept, focus on getting the concept. This is a concept that would require publicly available data to be executed. Phylo can now go ahead and then download the data sets that are relevant for what you want to do. And then, yeah, it takes it up from there, the whole downstream analysis. All right. So that is it that Phylo is capable of doing. So we can think broad, whatever is our interests. I don't know if I'm sharing my screen. So like I mentioned, Phylo can do so many things because we have so many skills available to it. So if you check here, you can explore Phylo's abilities or capabilities ranging from genomics, transcriptomics, epigenomics, proteomics, metabolomics, multiomics, functional enrichment, drug discovery, molecular design, experimental design, literature. So Phylo is able to have access to all these databases related to some of these analysis that you're going to be doing. All right. So I think that shouldn't be an issue for anyone who wants to do something. I think Phylo can do that for you, even if you don't know how to access the databases and download them. So we are talking about only open access data. All right. So if it's data that comes with
Okay.
restrictions, then I'm sorry, that cannot be done. Okay.
Yeah, I mean, just to also chime in, I think one of the things we do in grad school or maybe final year training is how do you identify research gaps? How do you identify research gaps? And I think there are now platforms, softwares, and websites to really identify research gaps. So as Humphrey showed, like you could look at this broad areas like transcriptomics, metagenomics of something of a disease, a pathogen, of response to disease and so on and so forth. But I think also one thing you could do like today, um, I mean, if you're taking if you're in grad school, you probably do this manually, which is to maybe read the paper into detail, check the methods,
Yeah.
check the conclusion, check the aims of the paper, and see whether the conclusions really matches the results, and trying to see what kind of questions, or what kind of research questions did they leave on the table. I know, most people or most research labs have been very exhaustive of the research analysis. But I think one thing that you could do even now, today, is to take a paper, right? Any paper that you're interested in, original research, and basically do like deep dive, maybe Claude, ChatGPT, and then ask, what are maybe one, two, or three, questions that could still be addressed with the available, data sets, or maybe with, similar, data sets, right? And, that would, probably, guide you in, first of all, collating really like a list of, possible things that you might be interested in, and we could maybe like, pitch, that ideas to your advisor, or maybe you could even implement it with the free credits that we have, instead of just giving out research questions. You would rather give out potential results, and how best to really expand. Right. So the same thing you would have done manually in grad school, put it in ChatGPT and see what I could generate one or two research questions and follow up in the final. So that's my 2 cents.
All right. Thank you so much, Sam. I think I don't know if we have any question online.
Yeah, so we would really encourage you to really look at this thing.
Yeah.
Like, it's one thing looking at it, listening to it, and so nothing really doing it or struggling with it. So we would really encourage you to really look at that. And hopefully we could organize a hackathon soon in Kumasi or in Accra, or maybe online again. You guys could come around and really do something together.
Yeah.
But it's very important to really get your hands in them.
Yes, so I think that is it for today. Thank you so much for joining. We really appreciate your time. We appreciate that you passed through to learn, and then to also interact with us, and even among yourselves. I think some people are sending DMs. So yeah, I think this session actually runs every Friday, 2 p.m. GMT. And yeah, we bring some special guests sometimes to handle some of the sessions. So be expecting a lot of guests. And then we have hands-on sessions like what we are doing today. So we are able to connect what we are doing to what people are doing in the diaspora. So we've had Finn, who works with the ARC Institute. We've had Ali. We've had Jocelyn pass through. We've had Samuel himself handles so many of the sessions, and we are being supported by Paz, Prof. Kwartin most of the times, and all that. So please do well if you want
And
to.
Ed Frame will be giving a speak to you.
Please, is that?
And Ed Frame.
Yeah, Ed Frame. Ed
Yeah, thank you.
Frame. Are we recording this? Eric, we need you. We need you. We need you so much to do what we do in a very much more better way. All right. So please, if you want to get in touch with us, join our WhatsApp community. We share news, updates, and opportunities on Claude, AI, ChatGPT, Biomni, anything that has to do with agentic AI. the news, the latest news. You have people put them up there, even on our website. You're able to catch the newest things on agentic AI. Yeah. So since you're interested and since you joined, we'll be glad to have you there. On YouTube, we have our sessions recorded over there, so you can actually have a look at some of our previous sessions. So we've been having this for more than 4 months now, but unfortunately, we've not made so many of the content open access, but then now we are making them open access. All right. So please follow us on LinkedIn as well, and then we'll be in touch. We look forward to the next session on Friday. Please do enjoy your weekend, and then we wish you all the best. We are looking forward to hear your feedbacks about what you're doing with Phylo, Agentic AI, or all the things that you are learning from the Friday sessions. All right, so we'll be glad to have them on LinkedIn. We'll be glad to have them on our WhatsApp community. Just feel free to communicate and chat. All right, thank you so much, everyone. And then I think we'll bring the meeting to an end officially. I wish you a very fruitful weekend and all the best. Thank you so much.